Hi everyone, welcome to Tarvos TNPSC Agricultural Officer coaching classes and myself Dr. Anand Srikumar, your faculty on plant breeding and genetics. Welcome all once again to another session. Today, unlike the regular classes, today we are going to discuss on a very important aspect of our exam, that is the areas that you students need to focus for getting maximum score in your examination. TNPSC has been asking questions regularly on selected areas of plant breeding. Okay, because most of when we analyze the previous year examinations, question papers of examinations, we can see that few units are given more importance compared to others. Okay, those units we are going to focus on those areas today because there should be a strategy to achieve the goal of an exam. Okay, to crack an exam means not just working hard, it is smart work. A smart working student can very easily substitute a hardworking person. So today's session is on which all areas you should focus for TNPSC. If you have only a limited time, you are at the last part of your revision sessions and you have a limited time, which all areas need to be covered more in depth, which all aspects of that subject area need to be given more focus. Such things are going to be discussed in today's session. And in crop improvement, your unit 5, according to our analysis by the Tarvos team, we have identified that unit 5 of crop improvement syllabus, okay, that covers nearly 13% of your paper. Okay, that means the most important area of your syllabus of crop improvement is your unit 5, which includes some plant breeding aspects, mainly the heterosis part, because this is the era of hybrids. Most people cultivate hybrids, you may know that. So the details on hybrids, then the advancements, because you need updations. So biotechnological advancements and such areas are also given more focus. And in plant breeding side, you see the principles, mainly the principles and the comparison of different plant breeding methods are given more focus. Okay, and most importantly, Tamil Nadu Agriculture University released varieties, or the popular varieties in Tamil Nadu is given, is having more significance. That also you need to focus, and the PPV&FRA Act, because as you may know, recently the Seed Act amendments are being a topic of discussion widely, and other seed related and agriculture related acts and the legal side is of important nowadays, because there are many loopholes that is to be changed. There are many corrections being proposed to the government, and those things are going on nowadays. So that area is also given an importance. So today we will be seeing specifically which all areas are specifically focused. I have just mentioned the broad areas. Okay, now we will be giving a deep dive into the more specific areas of these major sections. Hope that is clear. So this is, understand very importantly that this is a analysis video of, or the strategy video for students that is prepared based on nearly 6 years of examination analysis. Okay, previous 6 years of exam analysis. So this trend is being followed, and you need to keep your pace according to that flow or the area of importance. So we will be moving into that. And when you see the questions in the previous years, I already told you it is based on the previous 6 year question papers analysis. Okay, here you can see a blueprint that shows that the unit 5 is that important, and more than 20 questions consecutively. You can see more than 20 questions consecutively every year. It has gone up to 28 questions. See, that's why we have made a specific section, or a specific class session for explaining you the importance of Unit 5 in crop improvement. See here, I have prepared a pie chart also showing that which all kind of questions were asked from these areas. So mostly nowadays exams mainly has statement type questions and match the following questions. Okay, statement type, match the following questions. Earlier the pattern was direct questions. Okay, direct factual, direct questions. You will be given only direct 4 options. You can just tick the right option and go. But now the pattern is changing. There are more of statement type questions. What is the importance of these statement type questions is you need to have a clear idea of the concept. Earlier there used to be a scenario where the students used to mug up the multiple choice question books and go to the exam, but that will not work in the current scenario or current question pattern, because to answer statement type questions you should be thorough in your subject. Okay, so what is the importance here? Statement type questions gives you more I can't say it gives you, but it takes more of your time and needs in-depth idea of the subject. For example, there are 4 statements and the options, the A, B, C, D options will be 1 is correct, 1 and 3 is correct, 1, 2 and 3 is correct, only 4 is correct. Okay, if these 4 are the options, if a person knows that the first statement is right and doesn't have a clear proper idea about the other 1 or 2 statements, he can't touch that question, because the second option also contains the first statement 1, third question also has statement 1 along with the other options. So a person who can very clearly tell the truth or false of each and every statement only can select a option. So this makes the question paper more tougher, and this makes this causes more time loss. More of your time will be consumed in such questions. Similarly, match the following. It also takes time. It also takes more in-depth idea. Okay, 1 or 2 options may be same. Options in the sense matches will be same, and the other 2 matches will be confusing. So that kind of question paper pattern changes are in progress nowadays. So you should adapt accordingly. You should plan your studies accordingly. So see here, earlier you see this dark purple kind of questions, direct factual questions was covering nearly 45%. Now it has reduced to 35%, and more of 25% of statement type questions and 22% of match type questions have come in. So a person with good idea of subject only can move. That is the main thing. You should be a learner than a by-hearting machine. You should learn how it works. You should learn the concept. Okay, dive deeper into the subject. Okay, when you dive deeper into the subject, you will be having time shortage while you study. Right? That's why we have these sessions. Which all areas you should dive deeper? That idea you will get it from today's this master effect class. Okay, so now we will go into the major areas. This is a priority map that we have prepared for you guys. So here the list shown here contains different areas of this unit 5. Okay, this unit 5, and these areas are listed based on the priority. That means the first topics will be having more importance in your exam. Okay, and the lower ones will be having lesser importance. It doesn't mean that lower one is not required. It means that in previous years less questions were asked from those topics compared to the other ones. Now we will be going into each and every portions specifically. Okay, first of all, there are many questions from the Tamil Nadu released varieties or TNAU released varieties. There are TNAU is advancing with a large number of new varieties. There have been multiple varieties for mainly climate adaptation. Okay, because climate change is the most important scenario that is being discussed in the current agriculture scenario. So that is very important. Mainly the major crops like rice. The Tamil Nadu Agriculture University has already released CO59 rice variety, while the Adudure research station has released ADT 56. Okay, and if you see in pulses, you can see that Vamban 12 has been released, and that is also for this rice fallow cultivation and for the integrated cultivation. Then CTD1 of groundnut is released. Okay, that is also, as I said before, for climate resilience to adapt to drought conditions. Okay, and also in many other horticulture crops are gaining importance nowadays, because government is also boosting horticulture sector very much. There are many fruit crops varieties released, that is an avocado, that TDK2 variety of avocado. Okay, and many other vegetables, the co4 of tomato with an extended shelf life, and this one, the ash gourd PLR1, then bindi hybrids, the COH5 and chili CO5, many other varieties related to horticulture crops are now being released by TNAU and other ICR institutions, which are being cultivated in Tamil Nadu. Okay, you should have a clear idea about that. You should be preparing on that, either from the TNAU website or from any other sources. Tarvos itself gives you a clear idea regarding those released varieties, okay, in through its detailed sessions, and you should be very thorough on that. If there are hybrids also, you should be very clear, because some of the varieties and hybrids names are confusing. Okay, you should be very clear which are all the hybrids, which are all the varieties released. And in with respect to the major crops, you should also have an idea what the purpose of that variety is. Okay, now I told you regarding the tomato, the CO4 and this CO5, these are for extended shelf life. Okay, while CO59, this is also climate resilient, high yielding. That major varietal characters are also important. Okay, hope that is clear to everyone. Okay, the main research stations, the Adurai, the Coimbatore campus, okay, your Madurai, and many other Srivilliputtur, and all those research stations and their short names along with the varietal name need to be remembered with first priority. Okay, then comes the second one, that is heterosis and hybrid seed production. Heterosis, I would say, this is the most important area of plant breeding methods, because compared to the traditional breeding methods, now the situation is that population is increasing very fast, and to feed that population you need high yielding varieties or hybrids. Okay, the farmer doesn't care about purchasing a seed, okay, the value of the seed or something, but farmer is more concentrated on the produce volume. So hybrids are taking over in most of the cultivated areas, not only in Tamil Nadu and in many other states. So heterosis breeding is a very important area, and the seed production, how to do seed production of each and every important crop also is an important area to focus. Clear? And in this case, heterosis breeding is mainly done through different methods. Okay, hybrids can be developed in different methods, and for that the most commonly used method is a male sterility. Here, plant breeding method heterosis is connected to the principles of plant breeding, which includes male sterility, which is very important. I would say in the different plant breeding principles, you should focus mainly on self-incompatibility and male sterility. Okay, male sterility and self-incompatibility, their classifications, in which all crops they exist, which all crops they are used for hybrid seed production, all those things you should have a very clear idea. Okay, and then coming to the next important thing, that is the breeding methods. Okay, in self-pollinated crops mostly, because heterosis breeding is mainly done in cross-pollinated crops, even though they can be done in both crops. Heterosis breeding is majorly followed in cross-pollinated crops. While in coming to self-pollinated crops, the traditional breeding methods are being used, and here how you need to study these breeding methods is you should be able to compare different breeding methods. Comparison is very important. Just the studying the different steps in each and every breeding method is not important. You should know what is the difference between pedigree method, bulk and single seed descent. What is the key differences? Study those points which makes these different breeding methods unique. Okay, that is very important. Then comes heritability and genetic advance. Heritability and genetic advance are two important areas from statistical genetics part. Okay, because these two parameters control whether a character from a parent is inherited to the offsprings. How much that character is inherited from the parent to the offspring. So in a breeding method, these two parameters are very important. That is the fourth area of importance. Then comes the advanced molecular marker areas. Molecular markers here nowadays, this is the traditional breeding methods are being taken over, okay, or overstepped by advanced molecular level molecular assisted breeding methods. Okay, mainly the marker assisted selection, MAS. Okay, so the different molecular markers are used in these methods, and you should be able to differentiate between different molecular markers. Okay, different molecular markers, which are the dominant markers, which are the codominant markers, what are the strategy difference between each and every mechanism of these markers. You should be having a very clear demarcation. Clear? Then comes tissue culture and totipotency. Both are very related. Because of totipotency only you can do tissue culture. What is totipotency? Totipotency is the ability of a single cell of a plant to give rise to a whole plant. Okay, so totipotency and the scientists related to that are very important from Haberlandt onwards to different media scientists who have prepared different culture medias for different crops. Those are very important areas. Then comes the transgenic breeding side. That is your mainly when you say transgenic itself, you will be thinking about the BT cotton and other things. Other than BT, there are other different BT crops. Okay, your GM mustard, genetically modified mustard. Okay, and many other crops are transgenically produced, and that is also very important. GM mustard is a genome edited one. Okay, and this is transgenic. Okay, these are transgenics. And the other one I told you about, the GM mustard, this is a genome edited one. Okay, genome is modified there. Right. Okay. Then comes the breeding methods of cross-pollinated crops, and other than heterosis breeding, you have mass selection, recurrent selection. Okay, when all other methods are focusing on improving a line, recurrent selection methods focus on improving the population. As a whole, you improve the population. Okay, then synthetics and composites is another area you should focus on. Then, as I said before, when I was discussing about molecular markers, you can see which one? Marker assisted selection. Regarding the molecular markers, which are the different populations used for marker assisted selection, and which are all the populations used for QTL mapping. Okay, we call it mapping populations. You should be clear about the difference between each and every mapping population, how they are developed. For example, RILs, recombinant inbred lines, are developed by single seed descent method of breeding, while recombinant inbred lines, while the recombinant inbred lines are RILs, while backcross inbred lines are developed by backcrossing. Doubled haploids are by doubling using colchicine and other chromosome doubling agents. You develop those mapping populations, and how you develop these mapping populations are very important area. Okay. In BD Singh and AK Singh book, there is a picture depicting that different mapping populations and how they are produced. Okay, you can refer to that. In our classes also we discuss all these things, but I am giving you a idea. Okay. When this video can be useful for your final revision and for students who start their preparation in the later stage. They have less time. They don't have time to cover all the syllabus. They only have time to select the focus areas and study. Then this is a very important presentation to them. Hope this is clear. Then comes the plant genetic resources, or a PGR, and the institutions associated with that. In these areas, which are all the important areas? Different institutions or research organizations which are part of germplasm conservation. Secondly, the germplasms and their area of origin, primary and secondary centers of origin. Then comes the different conservation methods, in situ and ex situ conservation methods. Okay, and in that in situ conservation methods, you have gene banks, your different types of seed collections, active collections, working collections, base collections. Okay, how they are stored and used for research. Okay, these things are of importance, right? Then comes the variety release. How you can release a variety, which are all the tests to be done, which are all the steps to be followed. And in that, you should be having a clear idea about the flow chart from a proposal to the certification. Till certification, you should have a flow chart and the order, the exact order for releasing a variety should be very clear to each and every one of you. Because this can also come in such a way that you can arrange these different trials in the correct order. There are different trials involved in releasing a variety. They can ask you to arrange this different trials in the correct order. Such questions can come. Okay, and as I told you before, the legal side, PPV&FRA Act for conserving these germplasms are very important in today's scenario. So you should give focus on that. Then wide hybridization methods by which you cross very distantly related crops. Okay, these are advanced biotechnological methods. You should have an idea of that. And polyploidy also is doubling your chromosome number. Okay, chromosome manipulations mainly done successful in vegetatively propagated crops and flower crops, where vegetative part of the crop is more important. Clear? Okay, you can see watermelons, seedless watermelons. Okay, that is very common. If I say that example, every one of you will agree, because that is seen in market very commonly. Okay, so it's a 3x, it's a triploid. Okay, such many products of that kind are available, which is of prepared by chromosome doubling or using the polyploidy concept. Okay, our wheat itself, every most days we will have a chapati or a roti or something, right? And that wheat itself is a natural polyploid. Okay, so polyploidy is also an important method of crop improvement. Clear? Then speed breeding. This is an advanced area where you give good conditions. The conditions very precisely, you give the exact conditions for that plant to grow. Okay, so the plant performs much efficiently. So you can advance your generations in 1 year itself. You can go for multiple number of crops. Your crop duration will be reduced and you will get seeds in a faster level, right? Okay, and these are the areas of focus. In the right side, I have given you more detailed areas. Okay, even though we will be discussing more on that. I have given you the different, we have already discussed that, but for your further reference, there are more specific areas from which questions have been asked in the previous years. Hope this is clear to everyone. Now we will go to few areas which need specific targeted analysis. That is, in your heterosis, we already told that heterosis, okay, heterosis and then heritability, genetic advance concepts are very important. In that, there is few trap areas that most students will not focus on. Everyone will think that heterosis is a general phenomenon, but in heterosis itself, you have narrow sense. Sorry, I'm not telling heterosis. Sorry, in heritability itself, you have narrow sense heritability and broad sense heritability. Clear? And here, using narrow sense heritability to predict the performance and using of narrow sense heritability for selecting plants is a better idea compared to that of broad sense heritability. Keep that in mind. Then you can ask that what is the difference between these two, because variance in the genotypic level divided by the variance in the phenotype gives you the heritability, broad sense heritability, while the variance, additive variance divided by the phenotypic variance gives you the narrow sense heritability. So in breeding, when you select a plant for advancing to the next generation, we check heritability. If the plant particular character has high heritability in that plant, then you select that particular plant. Okay, you use that character for selection if that has a high heritability. I am specifying in depth that in that case you should go for narrow sense heritability for selection. Okay, narrow sense heritability is the major parameter that should be taken for selecting plants. Clear? Then you should know the formulas for genetic advance, heterosis and background recovery. Okay, genetic advance is how much the character compared to the parent, how much more advanced your next generation is. That is the genetic advance. Okay, the for example, it is basically given by the formula K sigma P H square. H square means your heritability. We already discussed about heritability. Then coming to heterosis, you have this formula comes for the mid-parent heterosis. You should be very clear about the different kinds of heterosis also. There is better parent heterosis called heterobeltosis. Okay, then economic heterosis, mid-parent heterosis. Okay, there are different types of heterosis and different formulas. That is also very important. Then comes another formula is the background recovery. Okay, because questions have been asked before. Okay, recurrent parent recovery. Backcross recovery is the recurrent parent recovery in marker assisted selection. When we will be discussing more when we discuss on marker assisted selection. When you use marker assisted selection to transfer few genes into a existing variety, for releasing that variety as an essentially derived variety, you should have a good background genome recovery. That means the original plant's genome should be almost conserved and only the few genes extra should be added to that. Okay, we check that by using the background recovery, or we call it the recurrent parent genome recovery. Okay, this is the formula 1 minus 1 by 2, 1 by 2 raised to n plus 1 into 100. Okay, we express that in percentage. Clear? Okay, I'm not going to explain you the different how you calculate it, how the data comes and everything. We discuss all that during our class. Okay, this is the strategy analysis session. Clear? So these are the important formulas that you need to focus on. Okay, one example is also given. Clear? And here the top trap, as I told you already, you should use narrow sense heritability for selection, or else what will happen? There will be higher estimates of breeding value and your selection goes wrong. Clear? Okay. Then coming to the male sterility system, that is also very important area. We also already discussed that male sterility and self incompatibility are two major areas of plant breeding principles you should focus on. Okay. Among the different principles in plant breeding, male sterility and self incompatibility are very important. And of that itself, male sterility is more important because it is used in hybrid seed production. So you should be very clear in the comparison between each and every male sterility systems, in which crops they are used, which is a 2-line system, which is a 3-line system. Okay, we should be very clear of which system is a 2-line breeding system, which system is a 3-line breeding system. Okay, and that comparison should be very clear. For example, there is a question comes and in an applied level, which of the following male sterility system is most widely used in red gram? In that crop level also you should be very clear which male sterility system is commonly used in which crop. Okay, for example, a red gram, the answer will be GMS. Okay, the genetic male sterility will be the answer. Similarly, for other crops also, for example, if rice comes in, that is CGMS. Okay, CGMS. Right. Then in the detail side of it, you have different lines in this hybrid seed production using the male sterility systems. That is A line, that is the male sterile line, the B line, which is the male fertile line, then the R line, which is the restorer line, which restores the fertility in the hybrid. Why we need to restore the fertility? You need seed production, right? Okay, you need to produce seeds, so that is also important. Okay, then I'm concluding. In male sterility, which all things you need to focus? Comparison of different breeding, the different male sterility systems. Okay, which are all the lines involved in each and every male sterility system? Which are of the major crops? Okay, which are all the major crops which are utilizing this male sterility systems in hybrid seed production? And the one more important thing, that are the different cytoplasm. There are many male sterility cytoplasms identified in different crops, and few of these male sterility cytoplasms are associated with some negative impacts. Okay, you should have a very clear idea. Okay, for example, Texas cytoplasm, Milo cytoplasm, Kafir cytoplasm. Okay, this Anand cytoplasm. Okay, and all these cytoplasms and their which crop they are found in, and what are the negative effect of this cytoplasm on other characters of the plant is also very important. You should make a table of that and revise it regularly. Okay, then one more thing to focus in that is which cytoplasm, okay, is present in which crop genus or species. For example, the CMS source of cotton, as mentioned here, CMS source of cotton is found in Gossypium harkensii. Okay, harkensii. No, not only this, there are more important areas. Okay, there are more important this male sterility source cytoplasms in main crops. Okay, in maize there are maize, sorghum, rice. There are many male sterility cytoplasms which are more important than this. You should know which species is having that particular cytoplasms. We make a table of all this and study well. Clear? Okay. Then comes the comparison of breeding methods. I already told you in that priority list we have seen all this. We are just focusing on in-depth areas. Usually questions comes from. Okay. Here mainly I told you when you study different breeding methods, have a clear comparison. Don't just study the protocol or the stepwise procedure. Instead, you select the important key points which makes each and every breeding method unique. Which is the fastest, which is the slowest, which needs more generation, which needs lesser generation, which is easier to handle. Okay, for example, single seed descent method is very easy to handle, but there is a defect that during this rapid selection of advancing few seeds, there is chance for losing good lines. Okay, for easy management, SSD is mainly used. Okay, which method help you to gain homozygosity in a faster way with fewer generations? Answer is single seed descent. Okay, you have a comparative graph in the lower side also. Okay, so in this way, compare each and every method. I have here, there is a comparison on the timing, speed, best for which purpose. Okay, and all things are given. We don't take this as a slide to study things. Okay, this is just a strategy video. I again repeat, you need to go for point by point comparison in your detailed in-depth classes and make notes of that. Clear? Okay. Then coming to the molecular markers, we already told this is in the top 10 priority section. Molecular marker, you should, as I told you, here also you should have a clear comparison. Okay, questions can come in different way. First and foremost, based on the type of molecular marker. Which of the following is a non-PCR based marker or a PCR based marker or an isozyme based marker? Okay, or a genotype, this SNP type genome level marker. Okay, such questions can come. And then which of the following is a dominant marker? Which of the following is a codominant marker? Such questions can come. The abbreviation of the marker will be given. For example, RAPD. Okay, or RFLP. Give the full expansion, restriction, fragment length, polymorphism. There was a previous year question based on SSR. SSR is simple sequence repeats. Simple sequence repeats, not single sequence repeats. Right? There was two options. One was single sequence repeats. Other one was simple sequence repeats. Answer is simple sequence repeats. Clear? Such minor things also you should focus. Okay, this is the third type of questions that can be asked. Then fourth, based on the applicability. Which of the following can be used for genotyping a large number of germplasms? Okay, or for a diversity study, which of the following is used widely? Most widely currently used is SSR markers. Okay, if RAPD is also there, you can also give that answer. More widely used is SSR. RAPD also can be used for diversity studies. Okay, such questions based on the applicability of the marker, which marker is for which purpose. Clear? And the last area from which the question can come is the techniques involved in this marker analysis. Okay, for example, blotting is involved in which of the following marker studies or which of the following molecular markers? For example, RFLP needs blotting, while SSR markers and SNPs does not need blotting techniques. Okay, such things that the technical differences of how this marker analysis performed, the key differences, that also you need to focus. These are the 5 areas of molecular markers from which question can be asked. Clear? When you study markers, search for these 5 points. Which kind of marker? Codominant or dominant? And what is the expansion of that marker? Then what is the applicability, purpose of using that marker? And what are the key methodologies like blotting and staining? Which are the key methodologies involved in that marker study? Okay, these 5 points you need to be clear about each and every marker. Hope this is clear to everyone. Okay, and then with these markers, you go for marker assisted selection. Marker assisted selection, that means using these markers, you are assisting, these molecular markers are assisting you in plant selection. For that, if you are going to search for only few genes, that particular gene, targeted gene, then that kind of checking for the presence or absence of a particular gene in a plant is called foreground selection. If you are using a large number of markers to check whether the whole genome of the plant is available in that particular line, then you call it background selection. During this background selection only, you go for recurrent genome recovery percentage. We already discussed that formula. Hope everyone remember that. Previous slide we have told about the RD, recurrent parent genome recovery percentage, and its formula. That is done during your background selection or with your background selection data. Clear? Okay. And for getting a clear idea, you should refer to the marker assisted selection based lecture. I have already made the videos of that. Okay, that is available, I think, for you people, or refer any other textbooks which contain marker assisted selection, which is a very important area. Okay, and the varieties, one more thing, varieties released using marker assisted selection. For example, the first rice variety released using marker assisted selection is Pusa Basmati 1. Those kind of extra points also you should be thorough with. Clear? Okay. Then comes the TNAU released Tamil Nadu state varieties which are being cultivated in the maximum level, and this is, I already discussed with you. This is the most important area, because out of this area there have been regular questions. At least you can expect 3 or 4 questions for sure from this state released varieties. Mainly from TNAU released varieties are commonly asked. Okay, there this edited Adudurai, when you go for example, if you take rice, the major research stations are Adudurai, then Coimbatore are the major, and Madurai also is one of the important stations, right? And which are all the major recent ones, which are the key or the most important ones? For example, MDU3, okay, is particularly resistant to gold midge resistance, having gold midge resistance. ADT45, which is a very common variety. ADT45, that is BPH, brown plant hopper resistant line. Okay, and similarly, I am not focusing on one or few varieties. Okay, you should be very clear with the recently released variety. Okay, you just study the last varieties which are released in the previous 2 to 3 years. Okay, I'm not asking you to skip all other varieties which are released before that, no, but before that period, the varieties released before that period needs less focus. Instead, you need to only focus on those varieties which were very popular. Okay, for example, this Gingerly, Srivalliputtur varieties, SVPR varieties are very popular. Okay, you need to focus on that particular genotypes. Those varieties we need to be clear, which is a white Gingerly, which is the black one. That major differences and the key or the most successful varieties which were used in very large areas, that you should be clear. And in addition to that, the recently released ones. This is how you study, not to give equal focus to all the varieties in the huge list. Okay, you have nearly, in rice, if you see, you have nearly 59 varieties from Coimbatore itself. You can't study the characters of each and every variety. This is not possible at all. Okay, for that, keep this strategy, last 3-year varieties and their characters, and the previously very much successful varieties which are cultivated in the large area also need to be studied. Clear? Yeah, here we have that Srivalliputtur VRISU2, that is a white sesamum. Then you have TVM3. I already told you regarding in the initial slide itself regarding few of the recent varieties from Tamil Nadu, which are being cultivated in Tamil Nadu. Okay. There are many hybrids also, but hybrids names are more easier to learn, okay, compared to the varieties. Okay, yeah. So you should be very clear. For example, MDU3 is resistant to BPH resistance. Here, MDU3 is Galmage resistant. Sorry, MDU3 is this Galmage resistant, not BPH resistant. But ADT45 is BPH resistance, not Galmage resistant. Okay, hope this is clear. Important. This is a very important area. You should not go to the exam without having a thorough idea about the varieties that are being cultivated in your area. Okay, in the previous 6 year itself, you see there are 21 questions. That means in an average 4 to 5 questions are for sure from the Tamil Nadu state varieties. Clear? Okay. Then coming to the next point, these are all the top 10 areas of that priority list. We are just giving a bit more deeper areas, important areas that you need to focus on. And regarding tissue culture, I would say that you should be knowing about the different medias and which scientist gave the media and for which crop. Okay, this is very important. That is given in that list also. You can see the Haberlandt in 1902, who identified initially regarding the totipotency, and he is called as the father of tissue culture in plants. Okay, then White successfully did callus culture. He developed White's media. Okay, then Skoog and Miller, they identified cytokine and different hormones which are used in tissue culture. Okay, and major contributions of the scientist in developing the different aspects of tissue culture is very important. Then the different growth hormones which articulate the success of tissue culture is very important, starting from auxins, cytokinins, gibberellins, abscisic acid, okay, and other brassinosteroids. Okay, or all these hormones, their scientists who identified them, from which organism they were initially identified, what is their purpose in tissue culture, what are the natural and artificial forms in which these hormones are available. For example, IBA and IAA, which of the form is natural, which is which is chemically prepared, which is a natural form. Such kind of questions are very important regarding the hormones, scientists, their contributions and different medias. Clear? This is about tissue culture side. For example, and other scientists, in the sense, you should be also clear about the clear regarding different kinds of tissue culturing. For example, Guha and Maheshwari, Andhra culture. Okay, there's an Indian scientist who did Andhra culture first. Such important points regarding scientists are very important in that area. Clear? Yeah. Then coming to transgenic breeding. This is also very important. Transgenic breeding, you already know regarding the BT crops. You should be very clear about the different BT genes. Some of the BT genes give resistance to Lepidopteran pest, while some other BT genes give resistance to Dipteran pest. Which genes give resistance to which category of pest? That is very important. Then the different markers, GUS markers, GFP, green fluorescent protein markers, the yellow fluorescent protein markers, different markers, luciferase marker. Okay, different markers which are used in transgenic breeding to identify the transformed lines or cells. That is important. Then regarding your Agrobacterium tumefaciens and the Agrobacterium rhizogenous species. Okay, what are the key differences? That is important. Then different transgene integration methods like gene gun, electroporation, okay, and agrobacterium mediated transfer, different methods, and the different hormones which are hormones or enzymes which makes transgenic breeding possible. For example, to make cuts in a DNA, you go for DNA, this one, polymerase. Okay, and for sealing the nicks or the cuts, you go for DNA ligase. Okay, sorry, polymerase is mainly for increasing the length. Restriction enzymes for cutting the DNA. Ligating enzymes like DNA ligase for again sticking the DNA part together. Okay, endonuclease enzymes and restriction enzymes are also very important. Clear? And you should be very clear about the different vectors also used in transgenic breeding, which are all the kind of vectors, which are all the chemicals involved in this transgene development, which are all the major markers used for transgene identification, which are the major transgenic crops, which are what are their main purpose. Okay, clear? For example, in tomato, you have different this transgenic types. Flavor saver tomato, which is having a higher shelf life. Okay, okay, such purpose also you should be very clear. Okay, hope that is clear. Then regarding the specific biotechnological methods like somatic hybridization, we already told when you fuse two different protoplasm of very distantly related organisms and develop a new crop species. Okay, tomato and potato together. Okay, you may have seen the pictures of such crops, right? Yeah. Then one more important point that is mentioned here is regarding your PCR. What is PCR? PCR is polymerase chain reaction, and PCR we already discussed about molecular markers. PCR is an equipment or a method your PCR or polymerase chain reaction is done in a machine called thermocycler. Okay, thermocycler where temperature can be increased and decreased very fast in a fast manner. So this is used for increasing the copy of the DNA you have. That is very important, and it was developed by one of the scientists called Carey Mullis. Okay, keep that point also in your mind. Okay, these are the important areas regarding tissue culture and transgenic crops, right? Then the another important area that we told was the PGR. In PGR, what are the areas to be covered? First and foremost, crop and their origin. Okay. Second, the organization involved in plant genetic resource conservation. In India, you have NBPGR, you have FRI, Forest Research Institute. Okay, you have many other organizations in India. In international level, you can see CGIAR institutes are there. Okay, then Biodiversity International, Rome is there. Such institutions are very important. Second, third one, the types of conservation of this germplasms, in situ and ex situ conservation, and its classifications. There are different kinds of in situ and ex situ classifications. Okay, conservation classifications, that is also very important. And which category, which type of crops are conserved in which method, that is also very important. In situ conservation itself, I already told you there are different types of collections: active collections, base collections, working collections. You should be very clear about the storage conditions of each and every collections. Your temperature, humidity, duration at which it can be stored. Okay, of different collections is very important. For example, base collection, you need to store at -20. It can be stored for more than 100 years. While active collection, you maintain it in the at 0 to 4 degree, just like your refrigerator. But you need to use it within 25 years. Okay. Similarly, you should be very clear about the humidity, temperature, and the duration of storage. Okay. Hope this is clear. And then last and final, you should be clear about the long stem storage vault. That is given in the slide also, that the Svalbard seed vault that is present in one of the island country in Norway side. Okay, India also has one. In Jammu Kashmir, we have a very small facility for conserving the germplasms for a very long time for your future, long future generations, or that we can say, even if some natural calamity happens and the whole world goes below the ground, these vaults will be having the seeds and all matter that is needed for starting life again. Okay, that's a very curious thing that also you should be having a very clear idea about the math. There are only very few points in that, you should be clear on that. Clear? Okay. Then comes the legal side, that is the PPV&FRA Act and the different test and the variety release procedures. Okay, yeah, for dust test, because distinct for releasing a variety, it should be distinct, uniform and stable. Okay, it is not a test for yield. Okay, dust test is not a test for yield, it is for a distinctiveness, uniformity and stability testing. Then the VCU test, and what are the purpose of doing all these tests, and the different varietal release protocols or steps you need to follow for releasing a variety also should be very clear. There is central seed committee, state seed committee, different types of varieties. You have extended variety, you have a multi-line variety, you have a pure line variety, and all those varietal ideas also should be very clear. Okay, in that itself comes the GI and other IP, the PBR. Okay, and the PBR and other this germplasm conservation, for conserving the germplasms, or and not for conserving, sorry, regulating the use of germplasms and conservation of few authentic type of genetic materials. You give some protection to them. Okay, like geographical indications, like patents, copyrights. There are many protections which can be given to a innovation or a technology or a crop variety or something like that. Okay, there are different categories of protections that also you should be very clear. That is the copyrights, patent, trademarks, geographical indications. Okay, this legal side also you should be very clear. Then heterosis, we already told, the heterosis is one of the most important category with respect to hybrid development. Okay, there are different methods of developing hybrids, that is single way cross, three-way cross, double cross, multiple cross, top cross, that is also very important, top cross. Okay, double top cross. How you represent those crosses is very important. There has been multiple questions in that area. That is very important, and different varieties are released through these different kinds of crosses. Okay, for example, there are many wheat varieties, okay, which are developed by using this heterosis concept. Ganga, Deccan, Rajat, there are multiple varieties released, and these varieties which kind of crosses were used for developing these varieties is a very important area to be studied. Clear? Then about this alien line, few terms like alien lines, bridging species, those definitions also you should be very clear. Okay, hope this is clear to everyone. Different heterosis, its category, types of crosses, and which are all the varieties developed by the different types of crosses. Clear? Yeah. And how you represent that also. Right. And then a few areas that are that you need to compare and contrast. I told you, while studying different areas, you need to compare the areas, identify the key points that make it mainly unique. You should be very clear. Here there is a list of few such comparisons that you should focus on: GMS and CMS, A line and B line. What is the difference? A line is male sterile, B line is male fertile. Like similarly, GCA and SCA, general combining ability and specific combining ability. Okay, while GCA is mainly focused on additive gene action, while SCA is for dominant gene action. That is the main difference. So you should be very easily comparing these different topics: RFLP and AFLP markers, SSR and RFLP marker. Okay, in-situ and ex-situ conservation, which are the different types of in-situ and ex-situ conservation methods. Okay, and such things you should be very clear, and this is just a priority list. whenever you study any method, if you find any way to compare it with any related method, you should be having that kind of approach while studying for competitive examinations. Okay. Now, because nowadays exams are linking multiple topics, okay, in one question. So your studies also should be in that line. Hope this is clear to everyone. Finally concluding your last minute revision, okay, when your exam is over your head, okay, you are going to write the exam in next 5 days or something, never skip these areas. The TN released varieties, once again repeating, heritability and genetic advance, molecular markers, conservation of germplasm, different crosses involved in heterosis breeding, okay, then male sterility, and also I would say that self-incompatibility is also very important, okay, male sterility as well as self-incompatibility, different breeding methods and their comparisons, okay, tissue culture and biotechnology, scientist names and the technology uses are very important. Then the legal side, the PPV&FRA Act and the IPR regulations. And when it comes to seed, there is Seed Act is also very important. Clear? These are the main areas that you should revise, and these areas can assure you a very good score. Okay, at least 20 marks. If one correct answer carries 2 to 4 marks, okay, usually there are negative markings also. So if you study these areas very well, okay, you will be getting 4 to 5 questions correct. Okay, so this is a very important area. The unit 5 of crop improvement is very important, and you nearly get 40 marks out of it. Okay, at least 4 to 5 questions is the bare minimum. 4 to 5 questions, and it can go up to, you have seen that, right? From each areas you are getting 4 to 5 questions, means in total your question paper is having more than 20 questions from unit 5 means, okay, means you are getting nearly, if you have for example 100 questions or 200 questions, 100 questions for say, no, you keep it 200 questions, then also you are getting 1/8 of it. If you are having 1/8 of it, or if you are having 100 questions, your 1/4 of your questions are from this particular unit. 1/4 of the questions from crop improvement is from your unit. Clear? From this unit 5 that we have discussed today. Hope these things are clear to everyone. When you don't go to exam without unit 5, without unit 5 covering the specific areas that we have discussed today. Most importantly, just keep a screenshot of these priority map in your phone. Whenever you take unit 5, cover these areas first. Clear? Hope this session was useful to all students. Okay, thank you for being with me, and we will be coming up with much more effective focus area discussions and other detailed class discussions in the coming days. Okay, hope everyone's exam preparations are going well. All the best to everyone, and prepare well, prepare in depth, in conceptual way. Don't just mug up things. Understand the concept, prepare well, and crack your exam. Okay, all the very best to everyone, and we will be winding up today's session, and we will meet in another session of strategy discussions and other class discussions also. Thank you everyone.